present or in the areas where the disease are not started yet. So we can exclude the pathogen. We can prevent the pathogens. We can delay the spread of the plant disease and pest. In the areas where disease is not started and pathogens are not present. So these are the objectives of exclusion. And we can exclude the pathogen by a number of methods and techniques. You can see here, we have to inspect whether the pathogen is present or not, especially in the propagating materials. You know, the propagating materials we call seed. It may be agricultural seed, it may be botanical seed. So first of all, we have to check the presence of pathogen in the seed. In the seed. And how can we do this? By testing the health of the seed. We call it seed health testing. There are a number of methods by which we can test the presence or absence of pathogen on the surface of seed, even inside the seed. So by inspecting the presence of pathogen on the surface or inside the seed, we'll issue a certificate. Let's say, for example, we have tested a seed lot, and we have got that out of 100 seeds, for example, we have got 5 seeds having pathogen. Then we'll issue a certificate that this seed lot contains 5% infected seed. It may be infected with fungi or bacteria, or maybe infested with nematode or other group of pathogen. And then we'll issue a certificate based on the testing result, the result of the testing, and then we'll issue a certificate. Okay. So this is a method we called inspection and certification. Let's say the seed lot we are testing is free of pathogen. That is 100% seed we call healthy. Maybe 5% seed might be infected. Then what happens? If there are 5% seed infected or infested with the pathogen, then we cannot allow the farmers to grow this 5% infected seed. Although this is not a huge amount, but you know, this is the propagating material. So this 5% seed will multiply after cultivation, and this will create tons of seeds, for example. So we cannot sow the infected seed, even it is 1%. What we have to do? We have to treat those 5% infected seed. We cannot separate the 5% infected seed from the seed lot. In order to treat the seed, we have to treat all the seed lot, both the healthy seed and the disease seed, although the amount is very minimum. Then after treating the seed, we can treat the seed by a number of ways, especially with the chemical seed treating chemicals. For example, Vitavax, Plantvax, Provax at a rate of 0.3% of seed weight. Then after treating, we expect that the pathogen present in the seed is already inactive or is already killed. Then we'll sow the seed in the field. We call it seed treatment. Okay. We can also eradicate insect vector. You know, when we talk about the management of plant disease, also plant disease, we don't think about only a plant pathogen. We also think about the group of insect pests, you know, from the Department of Entomology. We can eradicate the egg mass or other propagating materials of different insect pests by a number of ways, especially by chemicals. Then another method we call quarantine. I already discussed in the last class. You know, this is the legislative measures with the help of legal terms. We can restrict the movement of pathogen from the diseased area to the healthy area, from the area where the disease is present to the area where disease is not present. There are a number of quarantine methods, you know, exclusive quarantine, or we call it embargo. This is the total restriction of movement of agricultural commodities total restriction. We called it embargo. That is, will not allow importing seed or agricultural commodities from the foreign countries. We called it total restriction or embargo or exclusive quarantine. That is, the total restriction will not allow not a single seed, for example, from the foreign country. Then number 2, regulatory quarantine. That is, will allow import seed, but we have to test the seed, test the health status of the seed, and afterward we have to issue a certificate. Okay. Then after testing, if we see there are 10% seed infected, then we have to prescribe that. Before going to the farmers, these seeds should be treated with seed treating chemicals. And this is called regulatory quarantine. And there are some other methods, for example, domestic quarantine. That is, for example, blast disease of rice may be seen in Moyinsinh region, but may not be seen in Papna or Bogura or Khulla region. So what we have to do, the rice seed of Moyinsinh region should not be moved to the other region where the blast disease is not present. We call it domestic quarantine. That is the restriction of movement of agricultural commodities from the diseased area to the healthy areas. So these are the ways by which we can exclude the pathogen in the uninfected areas. And we have to set the standard of certification. We can set standard for all crops in general, and we can set the standard for specific crop or of specific disease we are dealing with. So we call it general seed certification standard. And let us have a look. Certification. For example, in the Indian subcontinent, for rice, for example, it's called by this fungus Neoposia horrida, and reduction, you know, the seeds,
লাইট তো দেখতেছি স্যার, কিন্তু আপনার কথা শুনতে পাচ্ছি না।
That is, you can also hear me. Okay.
স্যার আপনার কথা ক্লিয়ার আসতেছে না স্যার, কেটে কেটে আসতেছে। কি স্লাইড দেখা যায় বাট কথা বোঝা যাচ্ছে না কিছু।
So you can see this slide, right?
Your voice is not continuous, sir.
Okay, I can hear you clearly.
But we cannot hear you clearly.
আপনার কথা একটু পরপর ভেঙে ভেঙে আসছে স্যার।
I think it's a headphone problem or other issues.
Okay, just try to follow my lecture. You know, we are not living in the era of 5G, so you will have some problems. Uh, okay, so did you hear until this slide clearly?
স্যার এখন ক্লিয়ারলি শোনা যাচ্ছে মোটামুটি।
So somewhat better than previous.
Okay. So did you hear until this slide clearly before?
Yes,
So
sir.
I am continuing with
Yes,
the
sir, yes, sir.
I am continuing with the certification standard. You know, there are four types of seat: bidder seat, number two, foundation seat, number three, registered seat, number four, certified seat. So what is bidder seat? The seeds which are produced during the hybridization process by the breeders themselves. We call it breeder seed. And the offspring of breeder seed during multiplication process, foundation seed. And this multiplication continues. So the offspring of foundation seed registerly, the offspring of registered seed during the multiplication process, we call certified seeds. And we have to set standard for different seed, for foundation seed, for breeder seed, for registered seed, for certified seed. So this is the ultimate seed. So you see here for the paddy, that is for the rice, if the disease is burnt, for example, position standard for foundation seed is 0.1%, and the certification standard certified seed for rice, when we're talking about the burn disease, it is 0.5%. You can see here. And this certification standard varies from disease to disease, from crop to crop. You can see here for sorghum, if the diseases are good, the certification standard is 0.02% for foundation seed,
Sir, interruption is
0.04%
আপনার
for
কথা শুনতে যাচ্ছে না একদমই।
Sir, your voice is not clear.
Sir, sir, I have a question. Sir, are you using?
স্যার আপনার কথা কিছুই বোঝা যাচ্ছে না স্যার, কেটে কেটে আসছে।
just try.
Sir, I think there is a problem in your network, sir.
আরে রাউটারটা রিস্টার্ট দিলে কিন্তু হয়।
স্যার কি কথা বলতেছে একটু শোনা।
Sir, we are not hearing you.
I'm not talking to you. Then how can you hear me? Okay, I am talking the eradication of plant pathogen and other principles of plant disease management.
স্যার
So
এর আগে যে দুইটা স্লাইড
one of the
পড়িয়েছিল ওই
most
দুইটা কিছুই বুঝিনি।
what's your name, please?
স্যার শামসুন নাহার।
Samsung. Follow the slide. Samsung Nahar, can you follow the slide?
জি স্যার।
Okay. So what you did not understand? Can you tell me?
স্যার এর আগের যে স্লাইডটা ছিল ওটা থেকে মানে আপনার কথা কেটে কেটে আসছিল তো এর জন্য মানে বুঝতে পারিনি যে আপনি কি বুঝিয়েছিলেন তখন।
Sir, could you please repeat exclusive and regulatory quarantine?
I am telling you that is we are talking about the exclusion that is the legal restriction of the movement of agricultural commodities, especially the propagating materials, for example, seed. And we are talking about quarantine that is exclusive quarantine or embargo. It means of the movement of seed from the diseased tree to the.
Sir, still your voice is not clear, sir. This discontinuous voice is coming.
But it seems to me that the connection is okay.
Sir, your voice is being
No,
interrupted.
just you don't need to understand all my voice. That is, I am using the slide. Just follow the slide. So I am not telling you to hear me only. I am telling you to follow the slide as well. So exclusive quarantine means total restriction of pathogen. Import of seed from the diseased area to the healthy area. And regulatory quarantine means will allow the import, but we will test the health of the seed. We will issue a certificate and will prescribe that this seed, the disease seed, although the percentage is minimum, maybe 5%, and domestic quarantine means will not allow the movement of disease seed from the diseased area of the country to the healthy area of the same country. For example, maybe rice blast is very severe in Moymenshing district, but maybe there is no rice blast disease in Khulna district or in Chittagong district or in Rajshahi region. So what we have to do? We have to restrict the movement of rice seed from the Moymenshing region to the other regions where the blast disease is not seen. We call it domestic quarantine. And for this, we have to set standard. We have to set standard for certification process.
1, 2, 3 microphone testing.
We call general seed certification and specific seed certification standard. Specific seed certification standard. So specific, that is, it is very specific for a single crop or for a single disease. and general means for a group of crops, for a group of disease. You can check here the seed certificate standard, the maximum percentage seed infection standard you can see for different crops. Just look slide.
Setting certification standard.
And there are 4 types of seed, breeder seed, foundation seed, registered seed, and certified seed. And the certification standard for each seed is different. You see here, for foundation seed is 0.1%, but for certified seed 0.5%. So these are just some example of seed
So
certification.
what does it mean, sir? 0.10, 0.5%. What is the expression of this value?
0.1% and 0.5%.
স্যার এটার মানে কি যে ফাউন্ডেশন সিডের ক্ষেত্রে 0.1% যদি ইনফেক্টেড হয় তারপরেও সার্টিফাইড মানে সার্টিফিকেশন দিতে হবে নাকি, মানে এরকম কিছু কি স্যার?
Yes, that is, for example, you are testing the health status of foundation seed, okay? And let's say the health status of foundation seed might be 5%. That is, 5% seeds are infected. Then what you will do? 5% is now not allowed. Only 0.1% infection is allowed. You understand? The seed the farmers dealing with, if the infection rate or the percent disease seed is 10%, for example, then that 10% seed is not allowed for import or is not allowed for cultivation.
জি স্যার, বুঝতে পেরেছি।
So if the maximum seed infection is 0.5%, only then we can allow that seed to be imported or to be allowed for sowing in the field. If it is, for example, 0.6%, then we cannot allow the seed to be imported or to be cultivated in the field. Okay, so this is the maximum percent infection for certified seed. Then the third principle we called eradication of the pathogen. Eradication means complete destruction of the pathogen from a particular area we are talking about.
Sir, from the previous slide, I have a question.
And
sir.
we can eradicate the inoculum, or we can eradicate. Yes, tell me. What's your question? Tell me. Please tell me your name.
During import of any seed from other
And
country,
then
what is the
ask
standard?
the question.
So what is the maximum value, like certification seed 0.5%, but during import of any seed from other country, what is the maximum value in that case?
Hello? Set standard for all crops, for all disease, especially for the quarantine pathogen. There is set standard in the quarantine station, and the quarantine officer will check that standard, and it varies from crop to crop, it varies from seed to seed, and it varies from disease to disease. Okay, operation standard for each disease of each crop. if the disease is quarantine.
Sir, I
Disease
didn't understand
management,
anything, sir.
we
Your
call it
voice
eradication.
is continuous.
So
স্যার
that's
শোনা যাচ্ছে না কিছু?
Sir, we are not hearing you clearly, sir.
can you follow the slide?
Yes,
স্যার
yes,
আমরা
sir.
কি স্যার ক্লাসটা স্যার পরের দিন কন্টিনিউ করতে পারি স্যার? আজকে স্যার প্রচুর সমস্যা হচ্ছে।
Sir, you better leave us today, sir.
আমরা স্যার পরের দিন কন্টিনিউ করি স্যার, আজকে স্যার আসলে সম্ভব হচ্ছে না স্যার এভাবে।
আজকে থাক স্যার।
স্যার আজকে অফই দেন, আপনার একটা কথাও বুঝলাম না।
অ্যাটেন্ডেন্স শিটটা দিয়েন স্যার।
So I can see that there are 265 students present at the moment, and I will continue until 5 to 10, and I am talking about eradication of pathogen. So eradication means complete destruction of pathogen from the particular area or from the diseased area. We can eradicate the pathogen by a number of ways. First of all, you can see here, this is a lengthy process. We cannot eradicate the pathogen one season, for example. We need several seasons. So we can exploit different beneficial fungi, bacteria, and nematode to control harmful fungi, bacteria, nematode, and even insect, for example. We call it biological control of the pathogen. Then, of the same crop, years after years, increase the incidence and severity of the disease. If we cultivate the same crop in the same field continuously, years after years, then the incidence and severity of the disease will be increased. So what we have to do? We have to rotate the crop. So if we cultivate rice, for example, this year in a particular field, then the next year we have to cultivate wheat, for example, or that is, we have to cultivate the non-host crop of the pathogen we are dealing with. We call it crop rotation. So by crop rotation, what happens? The pathogen become vulnerable. Pathogen don't get the host. In absence of the host plant, the pathogen cannot survive. So in this way, we can eradicate the pathogen from a particular field. Okay. So, and we can do this by a number of ways. Rogging, that is the destruction of the diseased plant from the field. Then we can eradicate the alternate host and collateral host. You know about alternate and collateral host from level 2. Then we can go for sanitation. That is, after harvesting, there are some plant residues or crop residues still remain in the field. So the pathogen can survive in the plant residues after harvesting. So we have to clean those remaining plant part after harvesting. We call it sanitation. Then we can cover the seed bed, for example, with the polythene. We can follow mulching. What happens through mulching and polythene covering, the temperature increased in the seed bed, for example, moisture will be very high. So in presence of high temperature and high moisture, the propagule, the resting propagule will be germinated. And when the resting propagule germinated, they produce hyphae and mycelium, and these are very susceptible structures. So in presence of high temperature, those structures will be killed. Okay, so by this way we can eradicate pathogen, and we can also deploy chemical treatment and heat treatment. We can also treat the soil by a number of ways. For example, with formalin solution, 5% formalin solution. So we can kill the pathogen present in the soil or in the diseased field. So these are the ways by which we can eradicate the pathogens. But this is a lengthy process. We cannot eradicate the pathogen overnight. It takes time. So by following these techniques, we can eradicate the pathogen from a particular field.
স্যার
Yes.
স্যার হচ্ছে অ্যাগ্রোনমিতে আমাদেরকে বলতেছে হচ্ছে অর্গানিক ম্যাটার অ্যাড করার জন্য যে পূর্ববর্তী যে আমি ক্রপটা করছি তার রেসিডিউটা মাটির সাথে ইনকর্পোরেট করার জন্য। প্ল্যান্ট প্যাথোলজি ডিপার্টমেন্ট বলতেছে সেটাকে স্যানিটাইজ করার জন্য। তাহলে অ্যাকচুয়ালি কি করতে হবে স্যার?
Okay, organic matters means you see sometimes we cultivate dhoinsa in the field, green manuring crop. So this dhoinsa normally cultivated to add organic matter in the field. Okay, but I am talking about the plant debris. That is, let's say we are talking about the rice field. We are talking about the rice disease. So after harvesting the rice, still there are some material. There are some debris in the plant.
স্যার অ্যাগ্রোনমিতে বললো যে আমরা রাইসটাকে পুরোপুরি কাটবো না, কিছু মাটি থেকে কিছু অংশ রেখে আসবো এবং ওইটাকে মাটির সাথে ইনকর্পোরেট করে নেক্সট আমরা হচ্ছে নেক্সট ফসলটা আমরা করব, নেক্সট যে ক্রপটা আছে ওটা আমরা কাল্টিভেট করব। তো সেক্ষেত্রে শুধু ধনচার ক্ষেত্রে বললো না, যে কোনো ক্রপ রেসিডিউ আমরা পুরোপুরি কেটে আনবো না, কিছু রেখে আসবো যাতে আমার অর্গানিক ম্যাটার কন্টেন্টটা বাড়ে।
Okay, that's also right, but it's applicable only when there is no disease in the field. We can also incorporate the rice residues, straw for example, if there is no disease in the rice. But if there is disease in the rice, then we cannot add the plant debris of rice to the soil. You understand now?
Yes sir, thank you.
Okay, so sanitation means it involves all the procedures that prevent the spread of disease.
স্যার লিংকটা দিলেন না তো স্যার?
স্যার ক্লাসের টাইম শেষ, অ্যাটেন্ডেন্স শিটটা দেন।
So there are 272 students present, right? And these 272 students, I will collect attendance in the next class. Tomorrow at 11 a.m. So see you on the next